saya terputus, terus call saya. Sebab I have no way of knowing. Okay.
So,
Okay, so semua dengar suara?
Dengar, madam.
Alright. Okay. Okay, so for today's yeah, okay. Okay, so for today's lecture, we are going to go through the chapter photosynthesis. Okay. Yeah, awal-awal ni saya akan speed through lah because it is just the normal information on photosynthesis. Okay. Alright. So these are the subtopics of photosynthesis, which is you are going to know the differences between autotrophs and heterotrophs. And then for 6.2, you are going to learn on the different types of pigments, photosynthetic pigments, where they are found, and what type of light, okay, that the pigment absorbed. And then in that, you are going to look at two different graphs, which is absorption spectrum and also action spectrum. Okay, so for these two graphs, you have to know what are the differences. Okay. Okay. So and then the next one, you are going to look at the two stages of photosynthesis, which is the light dependent reaction and the other one is dark reaction. Dark reaction ni lama-lama, so awak ignore je dark reaction ni. It is actually the standard way of knowing the dark reaction is Kelvin cycle. Okay, Kelvin cycle. So Kelvin cycle is actually the dark reaction which occurs in the stroma, which is the function of the Kelvin cycle is to receive carbon dioxide and convert carbon dioxide into sugar. But in order for Kelvin cycle to occur, it requires energy that is produced during the light reaction, which is in the form of ATP and also an ATPH. And then the last one, we are going to look at the difference between C3 plant, C4 dengan chem plant. Kenapa nama plant ni dinamakan sebagai C3, C4 dengan chem plant? Itu yang awak kena tahu. Okay. Kejap ya, ada yang nak masuk lagi. So, alright. Okay, so these are the learning objectives that you can go through on your own. Okay, so heterotrophs and autotrophs, ini biasa orang dah tahu kan. So these are the overview. Okay, so you know that carbon dioxide is the vital source to synthesize carbon-based biological molecule that make up the biomass of living organisms. So basically, all molecules, okay, they contain carbon. Okay, so and usually the carbon comes from carbon dioxide, and all life processes requires a continuous input of energy. Okay, so the sun provides most of the energy that powers life on earth. Okay, so basically the sunlight, okay, will be absorbed by the pigment chlorophyll of chloroplants for plants to carry out photosynthesis to produce sugar. Okay, so sugar or the cell wall of plants kan, so those are made up of sugars that are produced during photosynthesis that requires energy from the sunlight. Okay, so a self-sufficient ecosystem consists of physical environment inhabited by organism, produces, consumers, decomposes that depends on each other, and the environment for nutrients, energy, oxygen, and carbon dioxide. So all organism on Earth requires all these basic basic component lah kan, nutrients, energy, oxygen, and also carbon dioxide. Okay, so autotrophs are basically producers. They are organisms that carries out photosynthesis. Okay, so tumbuhan lah selalunya kan. So dia perlukan apa? Dia perlukan carbon dioxide, water and also light energy to synthesize organic compound which is glucose and other nutrients, and releases oxygen, okay, which is the vital byproducts, which is oxygen is required by other organism, okay, to carry out cellular respiration. So photosynthesis, examples of plants, examples of organism of producers, there are plants, okay, algae, certain protists. So what is protists? Protists is unicellular eukaryotes, okay, unicellular eukaryotes, certain bacteria and archaea. Okay, so these organic molecule produced by autotrophs can be used by both heterotrophs and autotrophs. Okay, so as food and also as the building blocks of cells. Okay. Okay, so examples of autotrophs yang nampak kat sini, terrestrial plants. Apa tu terrestrial plants? So they are land plants. Okay. So and then algae, purple sulfur bacteria, cyanobacteria and also multicellular algae. Okay. So heterotrophs, consumers. So they depends on food produced by producers and also oxygen. And then the other one that you have to know is decomposers. Okay, so decomposers are actually heterotrophs. They are consumers. And then the examples are bacteria and archaea and also fungi. So they break down. Okay, they break down organic materials from dead organism. Okay. Okay, so these are the energy flows. Okay, so these are energy flows. So the sunlight provides energy for photosynthesis to be carried out by plants, and then plant releases oxygen, and then it also produces foods, okay, in the form of glucose and also other nutrients, and also the cell wall of plants, okay, and then so these are used by heterotrophs, kan, nampak dalam gambar ni ada panda, ada lion and all other organism requires, okay, the oxygen produced by photosynthesis. And then, so the oxygen released from photosynthesis is used by these heterotrophs or consumer to carry out cellular respiration. Okay, so cellular respiration produces energy, kan, in the form of ATP, so that the ATP is used for organism to carry out life activities such as homeostasis, kan, growth, development, reproduction, movement of bodies, movement of materials across cells. Okay, and also the energy is used to synthesize other molecules and also structures. Okay, so these are the energy flow. Apa kaitan antara photosynthesis dengan cellular respiration? Awak kena tahu. So they are really depend on each other. Okay, so photosynthesis. So these are the definition of photosynthesis, which is the biological process that captures light energy and transform it into the chemical energy of organic molecules, which is carbon dioxide, which are manufactured from carbon dioxide and also water. Sekejap ya. Okay. So photosynthesis require oxygen, carbon dioxide and also water. Okay. So this is the overall chemical reaction of photosynthesis. It requires carbon dioxide, water, and also light energy. Okay. So the light energy is absorbed by the chlorophyll, and then the product is glucose, oxygen and also water. Okay. So kalau kita akan tengok kejap lagi yang photosynthesis ni dia ada dua stage, kan, light reaction and also dark reaction, which is the Calvin cycle. So bila masa air ni digunakan? Air ni digunakan masa light reaction. Okay, light reaction, which is light reaction tu ada satu proses kita namakan dia sebagai photolysis of water. Okay, so photolysis of water ni dia akan memecahkan air kepada hydrogen ion, electrons dan juga oxygen atom. Okay, so oxygen atom tu nanti akan menghasilkan oksigen. Okay, so kalau electrons tu, kenapa nak kena release electron? Sebab kita belajar dalam cellular respiration, electrons have high energy, kan, dia akan it has high energy, and the electron will transfer along electron transport chain and then releases energy to synthesize ATP. Okay, so ATP molecule ni diperlukan, ATP, dia akan menghasilkan masa light reaction ATP dan juga lagi satu NADPH. Okay. So NADPH ni macam cellular respiration. Dalam cellular respiration it is NADH kan, kita belajar. Okay, so dalam photosynthesis NADPH. Okay. Remember P bukan stand for photosynthesis, dia ada nama chemical dia. Okay. So these two molecules are produced during the light reaction. Okay, basically it is produced from the electrons. Okay, that release from the process of photolysis of water, and then it travels along ETC to release energy, and the energy is used to synthesize ATP, and another molecule is NADPH. So these two molecules will be used during Calvin cycle, and the Calvin cycle will consume carbon dioxide, and the carbon dioxide will produce glucose. Okay, during the Calvin cycle, it is to produce glucose. So glucose is actually produced during Calvin cycle. Oxygen is produced during light reaction. Water is produced during both lah kot. Okay. Tapi yang pentingnya kena tahu like the oxygen is released during light reaction, glucose is during Calvin cycle. Okay, so oxygen is the byproduct which is released during light reaction. Okay, so same as cellular respiration, so photosynthesis is also a redox reaction. Okay, so carbon dioxide will serve as the oxidizing agent. Okay, so oxidizing agent which is it accept electrons. Sekejap ya, ada sound effect kat belakang ni. Okay, so carbon dioxide will serve as the oxidizing agent and water as the reducing agent. Sekejap, ini adalah efek daripada kita work from home. Bear with me sekejap. Okay, so kalau tengok dekat equation photosynthesis ni, dia kata carbon dioxide as the oxidizing agent. Okay, so sebab apa dia akan menyebabkan water ni reduced. Kan, water ni akan reduce, maksudnya water ni akan kehilangan dia akan kehilangan hydrogen ion dan juga electrons. Okay, so water is reduced because carbon dioxide is the oxidizing agent. So water reduced, dia akan kehilangan hydrogen ion dengan electron, so dia akan jadi oxygen, okay, which is the byproduct of photosynthesis released during light reaction. Okay. Kalau carbon dioxide ini, it function as the oxidizing agent, so maksudnya dia akan reduced, mengalami reduction. Okay, reduce. So kalau reduce maksudnya dia akan terima hydrogen atoms lah, so dia akan jadi glucose. Okay, so ini awak kena tahulah kalau dalam cellular respiration awak kena tahu apa yang akan mengalami reduction, apa yang akan mengalami oxidation. Okay, so photosynthesis is endogonic process. Endogonic means that it requires energy, and the energy is provided by the sunlight. Okay. Okay, so ini adalah proses yang saya cakap tadi, splitting of water molecule. Splitting of water molecule occurs during light reaction. Okay, so water molecule will split into hydrogen and also oxygen, incorporating electrons of hydrogen into sugar molecules and releasing oxygen as byproducts. Okay, so ingat masa photolysis of water which occurs during light reaction, dia akan menghasilkan hydrogen ions. Okay, 2 hydrogen ion plus 2 electrons, sebab dia daripada hydrogen atom kat sini kan. So atoms akan menghasilkan hydrogen ions dan juga electrons. So because you have 2 hydrogen atoms, so it produces 2 hydrogen ions, 2 electrons and 1 oxygen atom. So in order to release oxygen molecule, okay, which is O2, it requires two water molecules that undergo photolysis of water, okay, to release one oxygen molecule. Okay. Okay, so this is the site of photosynthesis. So basically dalam sel daun ni, dia ada sejenis tisu, kita namakan dia sebagai mesophyll. Okay, mesophyll tissue which is consists of mesophyll cells. So mesophyll cell ni dia ada banyak sangat chloroplast. Okay, so it is the site where photosynthesis occurs. Okay, and within the mesophyll cells there are many air spaces. So kat sini yang dia akan accumulate carbon dioxide. So the carbon dioxide will enter the leaf through the stomata, and then the byproduct of photosynthesis, which is oxygen, will also be released from the stomata. Okay. So what are stomata? They are microscopic pores in the epidermis of leaf, allows the interior of the leaf to exchange gaseous to exchange gas lah, which is carbon dioxide and also oxygen. And then vein. Okay, so you have vein. So carries water from roots to the leaf, carry off sugar from the mesophyll cell to non-photosynthetic areas of the plant. So the leaf will carry out photosynthesis. Okay, photosynthesis produces sugar. Okay, so photosynthesis will basically occurs in the mesophyll cell. Okay, so once the sugar is produced, so they are carried by veins, okay, to other parts of the plant, such as the fruits. Okay, so fruits, as you taste fruits, they are sweets. Okay, they are sweet. And then roots, okay, tubers. So those are other parts of the plant that will store the product of photosynthesis. Okay. So chloroplast, okay, site of chloroplast. So chloroplast, as you know, it is organelle that carry out photosynthesis. So chloroplast has two membranes again. So the inner and the outer membrane, and the fluid inside the chloroplast we call it as the stroma. So stroma is the site of Calvin cycle. Okay, Calvin cycle. So yeah, suspended in the stroma is the thylakoids. Okay, so thylakoid is the third membrane of within the chloroplast. Okay, so it has thylakoid membranes. So the thylakoid membrane is where chlorophyll are embedded. Okay, chlorophyll are embedded. So the chlorophyll will absorb light energy from the sunlight. Okay, so you have stacks of thylakoid which are called as grana. Okay, grana. So arrange into stacks called as grana. Grana banyak-banyak stack. Kalau satu stack kita panggil dia sebagai granum. Okay, satu stack of thylakoids is granum. Okay, so banyak-banyak granum, then it is referred to as grana. Okay, 4 stacks. Okay, so inside the inside the thylakoids, you have the thylakoid lumens. Okay, so nanti thylakoid lumens ni dia akan terlibat dalam proses chemiosmosis. So kita dah belajar masa cellular respiration. Chemiosmosis adalah proses untuk menghasilkan ATP disebabkan oleh perbezaan concentration hydrogen ion. Okay, so in this case, kalau chemiosmosis dalam photosynthesis, kita ada thylakoid lumen. Okay, thylakoid lumen. Okay, kat luar ni stroma. So dalam proses chemiosmosis masa photosynthesis, dia akan menyebabkan hydrogen ion tu bergerak. Okay, so dalam thylakoid space you have high concentration of hydrogen ion. Dalam stroma kita ada low concentration of hydrogen ions. Okay, so dia akan menyebabkan hydrogen ion daripada stroma pump masuk ke dalam thylakoid lumen. Okay, so macam mana nak menggerakkan hydrogen ion daripada stroma ke dalam lumen, kan? Sebab dia daripada low ke high concentration, it is active transport. Okay, active transport. So basically it uses energy that is released from electrons. Okay, energy released from electrons. So nanti kita tengoklah macam mana proses tu berlaku. And then thylakoid membrane, some extend from one granum to another, so has pigment that absorb light energy at different wavelengths. Okay, so ini adalah chloroplast lah kan. So yang warna kuning ni adalah thylakoid lumen. So ingat thylakoid lumen will have high concentration of hydrogen ions. Okay, kalau stroma kan will have low concentration of hydrogen ions. Okay, so yang ini akan terlibat dalam proses chemiosmosis. Dia tak ada kaitan pun dengan dalam fotosintesis, kita tak tengok pun intermembrane space. So intermembrane space dalam mitochondria semasa cellular respiration dia terlibat dalam penghasilan ATP. Tapi kalau fotosintesis tak. Okay. So kat sini kita ada electromagnetic spectrum. So these are the range of different wavelengths, okay. That includes the very small, okay, the very small wavelength which is gamma rays. So kita belajar fizik kan. So kalau wavelength tu lagi kecil, lagi tinggi dia punya energy, kan. So kalau wavelength tu besar, so lagi rendah energy dia. So kalau untuk fotosintesis, kita hanya concentrate dekat visible light ni. Okay, so visible light, as you know, it consists of the range of wavelength that produce, okay, the 7 colors of the rainbow tu kan. So the 7 colors of the rainbow, ada 7 color tu, they have different wavelength and also energy. Okay. Kita take 5 dulu kejap. Okay, so light and photosynthesis. So visible light, okay, so it is the portion of electromagnetic spectrum that the human eye can see. Okay, so it ranges from 380 to 760 nanometer. So make sure setiap kali kalau nak tulis wavelength dia, you write the units. Okay, so this is the unit nanometer. Okay, so kalau tengok kat sini, okay, so 380 dia start dengan violet kan. So ingatlah color of rainbow. Sekejap saya nak ni. Okay, dia start dengan red, orange, yellow, green, blue, violet. So ingatlah dia punya color dia tu kan. So kalau saya adalah dia punya cara nak ingat dia kan. So you have your own methods. Okay. So Richard of York gain battle in vain. Tu secara saya ingat lah. Kalau bahasa Melayu ni saya tak tahu. Adalah yang korang ingat. Okay. So Richard of York gain battles in vain. So red, orange, yellow, green, blue, violet. Okay. So violet will have the smallest wavelength. Okay, which is 380 nanometer. So bila the shorter the wavelength, the higher the energy. Okay, so compared to red light, which is it has the largest or the longest wavelength. Okay, so the energy will be lower compared to the violet kat bawah tu tadi. Okay, so light is composed of small particles or packets of energy called photons. So bila dalam proses photosynthesis ni itu kata photons, it is light energy. Okay, so the energy of a photon is inversely proportional to its wavelength. So the shorter the light, the wavelength, okay, it has more energy per photon than longer wavelength. Okay, so violet 380 nanometer has nearly twice, okay, as much energy as photons of red light. Okay, so yang ni dia just nak bagi tahu kan. So kalau kita ada atom kat sini kan, so atom awak tahu dia ada electrons. Okay, so bila atom tu dia absorb energy, which light energy, which is photon, it causes the electrons of the atoms to be excited. Okay, to be excited, and then it can the electron can be released can from the atoms and causes the atoms to be ionized, and the electrons will be received by another molecule. We call it as electron acceptor molecule. Okay, so kalau dalam proses photosynthesis ni yang akan riliskan electrons kan, so basically yang akan riliskan elektron ada satu chlorophyll ni, kita panggil sebagai chlorophyll A kan. So chlorophyll A punya electron will be excited sebab fungsi chlorophyll adalah untuk absorb light energy kan. Ingat chlorophyll akan absorb light energy. So ada sejenis chlorophyll, kita namakan dia sebagai chlorophyll A. Bila elektron daripada chlorophyll A ni receive the photons, the electrons of chlorophyll A will be released and accepted by electron acceptor. So ini adalah permulaan dia masuk ke dalam electron transport chain. Okay, so chlorophyll A ni dekat dalam tilakot membrane tu nanti kita akan tengok dia ada dua jenis kan, dia ada dua jenis bergantung kepada wavelength of light yang dia akan absorb. Dia ada satu adalah P680 and another is P700. P680 ni adalah sejenis chlorophyll A, P700 ni pun adalah sejenis chlorophyll A. Okay, so bila dia absorb photons, dia punya electron akan will be in the excited state and then released from the chlorophyll A. So bila dia release, dia akan chlorophyll A ni akan jadi ionized, kan jadi P680 plus. Ini pun sama, P700 plus. Okay, so bila chlorophyll A ni dah release electron, dia dah bentuk ionized. So maksudnya macam mana photosynthesis ni nak berlaku secara continuous. So maksudnya electrons of chlorophyll A ni kena replace. Okay, kena replace. So mana nak replacekan electrons of P680 plus ni? Dia datang daripada electrons daripada photolysis of water. So sebab tu kena berlaku photolysis of water. Photolysis of water will produce electrons and the electrons will replace, okay, the missing electrons of these two chlorophyll. Basically P680 lah kan, P680 yang akan receive electron daripada photolysis of water, and then the electron akan ganti electron, akan dipas-pas-paskan sampai dia receive by P700 plus. Okay, itu yang akan berlaku masa the transfer of electrons, okay, masa light reaction. So photosynthetic pigments. So kat sini kita photosynthetic pigments are embedded in the thalakot membranes. This one awak kena tahu. Ini adalah antara soalan yang suka keluar dalam exam. So photosynthetic pigments and also electrons acceptor or electron carriers of ETC are embedded in the thalakot membrane. So that's why light reaction occurs and involve, okay, the thalakot. Kalau dalam soalan dia tanya where is the site of light reaction, awak boleh jawab dia berlaku dekat thalakot. Okay, so different pigments absorb light of different wavelength. Okay, so kita ada the most abundant photosynthetic pigments is chlorophyll. Okay, so the most abundant is chlorophyll A. Okay, the most abundant is chlorophyll A, or the most ataupun the main photosynthetic pigments ataupun most abundant is chlorophyll A. Okay, so for chlorophyll it absorb light primarily in blue and red light. Okay, blue and red light region ini awak kena ingat. Okay, of the visible spectrum and reflects green light So plant usually appear green. So kenapa daun kan dia appear green? Sebab the most abundant pigment is chlorophyll A. So chlorophyll A will not absorb green light. Okay, chlorophyll A will not absorb green light. So the green light will be reflected and transmitted. Ini awak tambah transmitted. Okay, reflect maksudnya kat permukaan daun awak boleh nampak dia warna hijau. Transmitted kat bawah daun tu awak boleh nampak dia warna hijau. Okay, so sebab the green light is reflected and also transmitted. So the wavelength yang dia akan banyak absorb untuk melakukan photosynthesis adalah blue and red light. Okay, so that is for chlorophyll A and also for chlorophyll B juga. Okay, so tapi chlorophyll B is not the main. Okay, chlorophyll B kita panggil dia sebagai accessory pigment. Alright. So all chlorophyll molecule in the membrane are associated with specific chlorophyll binding proteins. Okay. So nanti kita tengok kalau chlorophyll ataupun photosynthetic pigment dia bukannya free embedded in the thalakot membrane. Dia embedded in the thalakot membrane sekali dengan sejenis protein yang kita akan tengok kejap lagi. Okay. So that's why dia kata kat sini chlorophyll binding protein. Okay, so this is the structure of chlorophyll molecule. So dia ada profiling rings kat atas ni. Okay, so this is the region to absorb light. Okay, so beza antara chlorophyll A dengan B adalah dekat sini je. Dekat dia punya side chain tepi ni. Okay, so kalau chlorophyll A, dia akan ada CH3 metal group. Kalau chlorophyll B, dia ada CHO. Okay. So dekat ke tengah-tengah tu ada magnesium lah kan. So beza antara A dengan B ni je dekat dia punya side chain tepi ni. Okay, so the magnesium atom at the center absorb certain wavelength of visible light and reflects green light. So yang ini adalah hydrocarbon chain yang bawah ni. So it is extremely non-polar and enters chlorophyll to the thylakoid membrane. Okay. So we have here three different types of photosynthetic pigments. The first one is A, okay, chlorophyll A. It initiates light-dependent reaction of photosynthesis. Kenapa dia initiate? Sebab it is the main, okay, main photosynthetic pigment or the most abundant. Okay, the most abundant. So dia akan appears bright green. Okay, so this is the basically the color that it will reflect or transmit. Okay, transmit. So chlorophyll B. Chlorophyll B is known as accessory pigment. Okay, and also participate in photosynthesis. So dia akan appear yellow green. Okay, and then another pigment is carotenoids. So dia juga adalah accessory pigment. Okay, accessory photosynthetic pigments has various colors ranging from yellow to orange and sometimes can be red. Okay, yellow and orange lah. Alright. So sebab apa? Color ni dia akan reflect. Okay, reflect and transmit. Okay, transmit. So maksudnya for carotenoids, adakah dia akan absorb green light? So carotenoids akan absorb green light. Okay, so tengok dekat light spectrum lah, nanti kita tengok yang dekat sini. Okay. Okay, so kita kat sini kita ada satu instrument, we call it as spectrophotometer. Okay, so it is an instrument that measures the relative abilities of different pigments to absorb different wavelength of light. Okay, so ini awak kena ingat spectrophotometer. So kat sini kita akan tengok ada dua jenis graph, which is the first one is absorption spectrum. Okay, absorption spectrum of photosynthetic pigments. So ini adalah graph untuk absorption spectrum. Okay, so what is absorption spectrum? It is a graph of different wavelength of light that is absorbed by a pigment. So dalam absorption spectrum ni dia akan menghasilkan graph untuk setiap pigment yang kita nak study tu. So in this case chlorophyll A and also chlorophyll B. Juga boleh awak plotkan untuk carotenoids. So kalau tengok kat graph ni, Kan? So kalau kat graph ni dia combinekan both absorption spectrum and also action spectrum. Yang ni kita tengok untuk absorption spectrum dulu. So kalau kat sini digabungkan untuk carotenoids juga. Yang warna orange ni is the graph for absorption of carotenoids. So if you look at the graph, okay, so for carotenoids, alright, it absorbs from violet and also until green, kan? So itu carotenoids, okay. So dia absorb range ni. Tapi kalau chlorophyll A, kan, so the most light yang dia akan absorb is in the range of violet blue and also red light. Okay, so ini awak kena ingat violet blue untuk chlorophyll A and also red light. So yang ini adalah energy, light energy yang dia akan absorb untuk provide energy for photosynthesis. Sama juga kalau untuk chlorophyll B, kan, so dia akan absorb in the range of blue light, okay, and also apa kat sini, orange red macam tu, okay. Dia akan reflect light macam ni. So that's why, kan, kalau chlorophyll A dia akan appear yellow green. Okay, yellow green. Kalau chlorophyll A dia akan start reflect warna blue green tu, kan, tadi. Blue green. Warna apa tadi yang dia akan reflect tu tadi? Bright green. Okay, sorry, bright green. Dari kat sini dia akan start reflect kat sini, bright green kat sini. Okay, bright green yang dia akan reflect. Itu untuk chlorophyll A. Okay, so tengok chlorophyll A and B, okay, mainly absorb light mainly in the blue region and also red region. Kat sini, kan. So blue region, kalau untuk gabungan blue and chlorophyll A and chlorophyll B. So this will be absorbed. Dan sama juga kat sini will be absorbed. Okay, tapi range yang green light ni, green light will be reflect and also transmitted. Okay, so this suggests that blue and red light works best for photosynthesis. Okay, so green light is the least effective color. So kalau untuk action spectrum, it is an action spectrum of photosynthesis. Okay, so sorry, an action spectrum of photosynthesis is a graph, okay, of effectiveness of different wavelength of light for photosynthesis. So ini basically dia daripada action spectrum ni dia nak menunjukkan gabungan serapan light energy oleh kesemua pigment dalam daun tu. Okay, so gabungan serapan light energy daripada kesemua pigment dalam daun. So dia akan nak tunjuk, okay, rate of photosynthesis sebenarnya paling efektif dekat range of wavelength yang mana. Okay, so sama juga. So paling banyak akan absorb adalah blue and also red. Okay, paling sikit adalah green. Tapi kenapa ada absorption kat sini juga by green? Okay, for green light, sebab kat sini ada carotenoids. Okay, this is the range of green light that is absorbed by carotenoid also. Sebab tu ada absorption kat sini. Okay, kat sini. So an action spectrum shows the rate of photosynthesis at different wavelength of light. So to obtain the action spectrum, the rate of photosynthesis is measured at each wavelength for leaf, cells or tissue exposed to monochromatic light of one wavelength. So macam dalam apa, ada yang mini project buat, kan, rate of photosynthesis, dia tengok kesan different cahaya terhadap rate of photosynthesis. So boleh lah tengok, kan, kalau macam contohnya rate, macam mana nak tengok rate? Tengok how much oxygen bubble is released, contohnya. Ataupun you can see the growth of the plant. Okay, so maksudnya kat sini, okay, you are going to plant a seed of a plant, plant seed, and then awak pancar dia dengan cahaya warna hijau. Okay, compact tu bila awak pancarkan dia dengan warna merah kan, merah, dengan awak pancarkan plant tu dengan warna orange, dengan awak pancarkan dia dengan blue light. Okay, so antara keempat cahaya ni, mana yang akan show growth yang paling efektif, yang paling banyak kan? So kalau dia show yang paling growth yang paling tinggi for red light and also blue light, shows that the rate of photosynthesis is most effective under green light and also blue light. Okay, contohnya lah. Tapi bila awak pancarkan dia dengan green light, adakah growth tu tinggi juga? So ini awak kena buat eksperimen lah kan. So supposedly when you expose the plant to green light, the growth of the plant will be very slow. Okay. Okay, so macam tadi lah, so graph ni digabungan antara absorption spectrum and also action spectrum. So the action spectrum for photosynthesis is much broader than absorption spectrum of chlorophyll A. Kalau nak bandingkan lah kan, so action spectrum dengan absorption spectrum of one pigment. Okay, so obviously action spectrum is much more broader sebab dia hasil gabungan serapan semua pigment dalam leaf tu, okay, untuk melakukan photosynthesis untuk plant. Okay, so this is due to the presence of accessory pigments. So obviously dekat dalam leaf bukan ada chlorophyll A je kan. So dia ada chlorophyll A, dia ada chlorophyll B, dia ada carotenoids, dia ada much more. Sebenarnya photosynthetic pigment ni ada banyak lagi. Yang kita nak fokus 3 tu je, chlorophyll A, B dengan carotenoids. Ada banyak lagi sebenarnya, kita ada xanthophyll, ada banyak. Okay. So apa fungsi accessory pigments ni? So accessory pigment macam chlorophyll A dengan carotenoids is to absorb excessive light, okay, that would damage chlorophyll and also broaden the spectrum used for photosynthesis. Maksudnya kalau visible light spectrum ni kan, dia range from violet until red. Okay, so maksudnya dengan adanya accessory pigment ni, photosynthesis akan berlaku secara maksimum sebab ada banyak lagi pigment ni boleh absorb excessive light sebagai sumber energi tambahan untuk melakukan photosynthesis. Okay, so it broadens the light spectrum that the plant can use, okay, to absorb excess light to carry out photosynthesis. Okay, so itu fungsi accessory pigment. Alright. Okay, so photosynthesis kita nak tengok kat sini. It basically has two stages. Okay, the light dependent reaction and also the dark reaction. Dark reaction is also known as the Kelvin cycle. Okay, Kelvin cycle ni juga dikenali sebagai C3 pathway. Okay, light dependent reaction, location dia dekat thylakoid. Okay, dekat thylakoid. Okay, thylakoid, and to be specific, thylakoid membrane lah sebab it is where the chlorophyll pigments are embedded. Okay, kalau Kelvin cycle, the location occurs at the stroma. Okay, at the stroma. Okay. So okay, so kalau awak tengok kat sini, so this table just to differentiate between light dependent reaction and also carbon fixation reaction. Carbon, kenapa kita namakan dia sebagai carbon fixation? Sebab masa stage ni lah carbon dioxide will enter the Kelvin cycle, okay, and fixed. Okay, masuk fixed ni maksudnya dia akan combine. Okay, it will react. Okay, combine, react dengan another substance. Kita namakan dia sebagai carbon dioxide acceptor. Okay, so that's why kita namakan dia sebagai carbon fixation reaction. So dia sebenarnya adalah kalau paling common adalah Kelvin cycle. Okay, So light dependent reaction, it is the photo part of photosynthesis. Kenapa photo? Ini adalah stage yang mana light energy will be absorbed by chlorophyll. Okay, so carbon fixation, it is the synthesis part of photosynthesis. Kenapa synthesis? It is to synthesize sugar. Okay, Calvin cycle produces sugar. Okay, so light dependent, it takes place in the thylakoids. Kalau carbon fixation, it occurs in the stroma of chloroplasts. And then for light dependent, solar energy is converted into chemical energy. What are the chemical energy? It is ATP and also NADPH. Okay, ATP and also NADPH. Oxygen is the byproduct, okay, of light reaction. Okay, solar energy is converted into chemical energy, ATP and also NADPH. So ATP and NADPH will be used during the Calvin cycle. Okay. Okay, use the chemical energy, which is ATP and NADPH, which was produced during the light reaction to make sugar. So basically, Calvin cycle ni akan menghasilkan sejenis sugar, kita namakan dia sebagai G3P. Ingat lagi kan, dalam cellular respiration ada 3 carbon sugar which is G3P. So dekat photosynthesis pun akan menghasilkan G3P. Okay, Calvin cycle will produce G3P, glyceraldehyde 3-phosphate, a 3 carbon sugar. Okay, so in order to produce 1 molecule of glucose, the process requires 2 G3P. Okay, so this is to produce carbohydrates. Okay, carbohydrates. Carbon fixation produces sugar, which is carbohydrates. Okay, so for it says at the bottom here, for most plants, C3 plants, the carbon fixation reaction is known as the Calvin cycle or C3 pathway. Kenapa kita namakan dia sebagai C3 pathway? Ataupun plant yang carry out Calvin cycle ni sebagai C3 plant? Sebab the first product, okay, of carbon fixation is a 3 carbon sugar, which is dia ada G3P lah kan, G3P. Sebelum dia jadi G3P, dia akan jadi satu compound yang kita namakan dia sebagai PGA kan. So this is also a 3 carbon compound. Nanti kita tengok detail Calvin cycle tu. Calvin cycle akan menghasilkan first adalah molekul kita namakan sebagai PGA, 3-phosphoglycerate. Lepas tu dia akan convert jadi 1,3-bisphosphoglycerate sebelum dia convert jadi G3P. Macam tu. Dia ada reaction dia. Sama macam apa nama yang berlaku dalam cellular respiration lah kan. So it involve a series of chemical reaction. So remember, kalau photosynthesis, walaupun dia bagi equation sesimpel yang macam ni kan, sesimpel yang macam ni, macam saya cakap dalam cellular respiration, photosynthesis is not as simple as this equation. Okay, dia melibatkan banyak reaction yang perlu berlaku. Okay. Okay, so photosystem structure. So photosystem adalah komponen yang sangat penting semasa light dependent reaction. Okay, so photosystem adalah structure yang embedded. Okay, it is embedded in the thylakoid membrane. Okay, it is embedded in the thylakoid membrane. Alright. So a photosystem consists of antenna complex or light harvesting arrays, ataupun awak boleh panggil dia sebagai light harvesting complex. Okay, antenna complexes or light harvesting arrays or light harvesting complex. Sama je. Okay, so which consists of chlorophyll A, okay, chlorophyll B, and also accessory pigments. Okay, which dalam gambar ni represent by green circles. Okay, bound to protein, which is pigment binding protein. Yang warna purple ni lah kan. So warna purple ni adalah pigment binding protein. Okay, so another component of photosystem is that dia ada reaction center. So reaction center kat tengah ni. Okay, reaction center. Kat tepi ni adalah light harvesting complex. Okay, so light harvesting complex dia ada accessory pigment. Dia ada chlorophyll A, dia ada accessory pigment B, chlorophyll B and also carotenoids. Okay, carotenoids. So dekat tengah kita ada reaction center. So reaction center are surrounded by antenna complexes, consist of a pair of chlorophyll A. So these are a pair of chlorophyll A, warna yang diamond ni, yang diamonds, the green diamonds, proteins and also primary electron acceptor. So dekat okay, dekat reaction center ni kita ada primary electron acceptor kat sini. Okay, so this is primary electron acceptor. Okay, so itu adalah reaction center. Ingat reaction center ada a pair of chlorophyll A, proteins. So the chlorophyll are embedded in proteins, kan? And also primary electron acceptor. Sebab apa? Bila chlorophyll A ni dia absorb light energy, absorb photon, okay, dia punya electron will be excited. Okay, electron of chlorophyll A that is excited will be released and accepted by primary electron acceptor. So ini adalah permulaan electron transport chain dalam photosynthesis. Okay. Ya, ini sebenarnya dia keluarkan sebenarnya. Kalau kat gambar ni dia keluarkan ke atas. Sebenarnya primary electron acceptor adalah sebahagian daripada reaction center. Okay. So ini basically adalah photosystem. So photosystem dalam light reaction ni dia ada dua jenis. Kita ada photosystem 1 and photosystem 2. Okay. Okay. Photosystem 1. PS1 bukan PlayStation tapi Photosystem 1. Sekarang PlayStation nak yang apa? PlayStation yang kelima nak keluar ya. Alright. So and then Photosystem 2 ataupun PS2. Right. So dalam Photosystem 1 dia punya dekat reaction center dia ada sejenis chlorophyll A. Okay, kita panggil dia sebagai P700. So P700 ni is a pair of chlorophyll A. Kenapa kita panggil dia P700? Sebab chlorophyll A ni yang ada pada reaction center tu dia absorb light which is at 700 nanometer. 700 nanometer warna apa? Warna red kan? Alright. And then sama juga kalau photosystem tu dia punya dekat reaction center dia ada sejenis chlorophyll A, kita namakan dia sebagai P680, P680. Okay, sebab apa? Sebab dia akan absorb light at 680 nanometer. Okay, so bawah sikit pada 700 lah tapi still red light juga. Okay. Okay, so two possible routes for electron flow during light reaction adalah non-cyclic electron transport, okay, and also the other one is cyclic electron transport. For non-cyclic electron transport, dia melibatkan kedua-dua photosystem 1 and also photosystem 2. Dan yang ini awak kena ingat, for non-cyclic electron flow, dia akan menghasilkan kedua-dua ATP dan juga NADPH daripada light energy. Tapi kalau cyclic electron transport dia hanya melibatkan photosystem 1 dan hanya akan menghasilkan ATP molekul. Yang ini mesti dan wajib ingat. Okay. So kenapa kita namakan proses ini sebagai non-cyclic yang pertama kan? Non-cyclic sebab kalau untuk non-cyclic okay, non-cyclic maksudnya elektron tu asalnya daripada, walaupun kat sini kan, walaupun dia start dengan photosystem 1 tapi sebenarnya proses tu start dengan photosystem 2 dulu. Okay. Photosystem 2 yang akan start dulu. Okay. Lepas tu baru photosystem Lepas tu baru photosystem 1. Okay. So PS1, okay, dia punya chlorophyll A akan release electron. Electron will be transferred along electron transport chain and received by PS1. Kan. Lepas tu from PS1 dia akan the electron akan received by NADP plus molecule becomes NADP NADPH molecule. Okay. Semasa electron travel daripada PS2 ke PS1, energy akan released. Okay. And used during chemiosmosis. Okay. To produce ATP. So that's why kalau non-cyclic, maksudnya elektron tu asalnya daripada photosystem 1, dia tak balik semula dah. Dia terus diambil oleh NADPH. Okay. Terus diambil oleh NADPH dan juga elektron tu akan semasa dia travel along ETC, dia akan release energy untuk chemiosmosis berlaku untuk hasilkan ATP. Tapi kalau untuk cyclic electron transport, dia hanya melibatkan photosystem 1 kan. So photosystem 1 dia akan travel daripada photosystem 1, dia akan ikut kat sini. Dia akan balik kat sini, pergi kat sini balik. Balik, pergi kat sini balik. So it is a cycle. Okay. So semasa electron tu travel in a circle, so dia akan release energy and energy is used during chemiosmosis to produce ATP. So that's why for cyclic hanya akan menghasilkan ATP. Dia tak masuk kat bahagian yang NADPH ni. Okay. So kenapa walaupun dia start dengan photosystem 2, tapi dekat kita namakan photosystem 1 ni dulu. Sebab apa? Dia daripada saintis yang memula discover photosystem ni. Saintis memula dia discover photosystem 1 dulu. Lepas tu baru dia jumpa photosystem 2. Sebab tu kita namakan dia sebagai photosystem 1 and photosystem 2 walaupun photosystem 2 yang akan berfungsi dulu. Macam tu. Okay. Okay, so before kita proceed, ada soalan tak nak tanya? Untuk light reaction ni. Any question?
No, madam.
Tak ada Kita break 2 minit sekejap. Okay. So kita akan sambung semula. Alright, so now we are going to look at the first stage of light reaction, which is non-cyclic electron transport. Okay, non-cyclic electron transport. Ada buku dia juga panggil stage ni sebagai non-cyclic photophosphorylation, sama je. Okay, non-cyclic electron transport or non-cyclic photophosphorylation. Okay, kenapa dia panggil sebagai non-cyclic photophosphorylation? Sebab masa stage ni light energy akan digunakan. Light energy in the form of photon will be absorbed by the chlorophyll pigment, and in the process also involve phosphorylation to occur. What is phosphorylation? The formation or the production of ATP. Okay, from the process chemiosmosis. So basically chemiosmosis occurs during light reaction. Okay, so kita start dulu dengan PS2, okay, photosystem 2. Walaupun dia nama photosystem 2, dia akan start dengan photosystem 2 lah dulu, alright. Photosystem 1, sebab apa dia nama photosystem 1? Sebab dia di-discover dulu. Okay, berbanding dengan photosystem tu di-discover kemudian, okay. Dalam photosystem 2 ada P680. Apa dia tadi? Dia adalah special pair of chlorophyll A. Yang ni, yang diamond tu. Okay, so ini adalah reaction center. Reaction center ini. Okay, reaction center. Okay, so reaction center, it has P680. Okay, so photosystem 2 is activated when a pigment molecule in an antenna complex of photosystem 2 absorb a photon. Okay, so photon will be absorbed by accessory pigment in the antenna complex. Okay, antenna complex. So the photon will be transferred from one pigment to another pigment until it reaches the reaction center of photosystem 2. It is absorbed by P680. Okay, so the energy is transferred from one pigment molecule to another within the antenna complex until it reaches the reaction center. This transfer of energy is called resonance. Okay, so in the reaction center, the energy reaches a P680 molecule, which is P600 molecule is a special pair of chlorophyll A. Okay, so the energy excites an electron of P680. P680 becomes P708 plus. Maksudnya dia dah oxidized. Means that the electrons will be released from P680. Okay, electron P680 will be released sebab apa? It is excited due to the absorption of photons. Okay. So the electron is then rise to a higher energy level. So the electron is accepted by a primary electron acceptor known as pyrophytin, which is a highly modified chlorophyll molecule. Okay, so it is another type of molecule. Alright, chlorophyll molecule. So ingat primary electron acceptor duduknya adalah dalam reaction center juga. Okay. Okay, so the primary electron acceptor then passes the activated electron along electron transport chain until it is donated to P700 plus. Okay, P700 plus in the photosystem 1 and becomes bila dia receive electron, so dia maksudnya dia will be reduced, so dia akan jadi P700. So photosystem 1 dekat reaction center dia ada P700. So P700 ni sebab dia nak receive electron daripada photosystem 2, maksudnya dia dalam keadaan asalnya oxidized. Okay, so bila dia receive electrons kan, so dia akan becomes reduced jadi P700. Okay. So the electron connects, the electron transport chain connects photosystem 2 to photosystem 1. So as the photon travels along the ETC, electron loses energy, releases energy, then so releases energy, and the energy released is used to produce ATP by chemiosmosis. So the production of ATP is through chemiosmosis. Okay, chemiosmosis. So ATP production of ATP by chemiosmosis. Dah tulis kat sini, right? Okay, so ETC consists of several electron receptor in a series. So dekat sini sebenarnya ada a few more electron receptor. Okay, a few more electron receptor. Nanti kita tengok apa nama dia. Ada yang nama cytochrome, kan? Cytochrome, ada nama plastocyanin and ubiquinon. So those are the electron acceptor of ETC during photosynthesis. Okay. Cytochrome, ubiquinon, and then another one is plastocyanin. Okay, plastocyanin. Okay, kalau short form nanti awak tengok dia short form cytochrome ni CYT, ubiquinon PQ, plastocyanin PC. Okay, so those are the electron carriers of electron transport chains that are embedded also in the thylakoid membrane. Okay, so kenapa tengok kat dalam gambar ni, kenapa arrow warna merah ni dia menurun? Sebab nak menunjukkan electron initially it has high energy. It is highly energized electron. But as the electron is transferred from one carrier to another carrier to another carrier, the electron releases energy, and once the electron reaches photosystem 1, electron tu it has no energy left. Okay, no energy left. Bila electron tu di-receive oleh P700 plus to become P700, electron of P700 ni has low energy. Okay, so how to excite the electron of P700? So sama macam proses kat photosystem 1, dia akan absorb photon juga. Photon will be transferred from one accessory pigment to another accessory pigment until it reaches the reaction center of photosystem 1. Okay, so sebab tu nampak kat sini arrow dia ke bawah, sebab nak menunjukkan energy of electron will be released. Okay, and then once the electron reaches P700, the electron has no more energy, and in order to energize back the electron, the accessory pigment has to absorb energy and transfer it until it reaches the reaction center of photosystem 1. Okay. Okay, so activation of photosystem 1. Yang ni bahagian yang seterusnya lah. Yang saya baru explain ni tadi. So a pigment molecule in an antenna complex of photosystem 1 absorb energy. Okay, photon. Okay, absorb photon. So photon will be absorbed by accessory pigment, transferred from one pigment to another pigment to another pigment until it reaches reaction center of photosystem 1 and received by P700. Okay, so this is to excite electron of P700. So once electron of P700 is excited, P700 will release its electron and receive by the primary electron acceptor. So bila dia release, dia akan jadi balik P700 plus lah. Dia akan jadi oxidized, kan? Okay, oxidized. Oxidized release electron. Release kepada siapa? Primary electron acceptor yang ada pada reaction center of photosystem 1. Okay, so point number 2, the energy is transferred to the reaction center and reaches a P700 molecule. Ingat ini adalah a special pair. Ingat pair dia berdua of a special pair of chlorophyll A. Okay, of in the reaction center of photosystem 1. So the energy re-energized an electron of P700 So the energized electron is transferred to a primary electron acceptor, which is a special pair of chlorophyll A molecule also. Ini salah ni, ini bukan apa ambil je sampai sini je. Okay. Sampai sini je. So the energized electron is transferred to a primary electron acceptor. So the P700 becomes P700 plus. Maksudnya dia dah oxidize lah. Okay, sebab apa? It loses an electron. Okay. When it loses an electron to the primary electron acceptor. So ingat ya, bila the plus sign maksudnya it is oxidized, it loses electrons. So the electrons will be transferred along electron transport chain and received by ferredoxine. Okay. Ferredoxine akan transfer electron to NADP plus becomes NADH. Macam mana nak dapat H ni? Dia dapat H daripada hydrogen ion from the stroma. Okay. From the stroma, from the medium. Okay. So maksudnya NADPH molecule dengan ATP yang produce masa dia travels along ETC from photosystem 2 to photosystem 1, the production of ATP and NADPH ni will be released in the stroma. Sebab apa stroma? Sebab stroma adalah site untuk Calvin cycle. Okay? Calvin cycle. Calvin cycle akan menggunakan product of light reaction to produce sugar. Okay? Okay, so the energized electron is passed along ETC and to ferredoxine. Okay, ferredoxine. So ferredoxine transfer the electron to NADP plus, catalyzed by the enzyme ferredoxine NADP plus reductase. Okay, reductase. Dia akan reduce reductase. Sebab apa dia akan reduce NADP plus becomes NADPH. Okay, so only a single electrons pass down the ETC, but 2 electrons are needed to reduce 1 molecule of NADPH. The transfer of electron kan along ETC ni, dia akan transfer 1 electron at a time. Okay, 1 electron at a time, tapi dia perlukan sebenarnya 2 electrons. Sebab tu kat gambar ni dia just summarizekan sebagai 2 electron kan. Tapi sebenarnya dia akan travel 1 electron at a time. Okay, 1 electron at a time until it is received by NADP plus. Kenapa dia perlukan 2 electron? Tengoklah dia punya formula kan. NADP plus becomes NADPH kan. So kat sini plus sign, asalnya sini ada plus sign. Dia bergabung dengan H plus kan. So this one is missing an electron. This one also missing an electron. So 2 electrons are required to produce 1 molecule of NADPH. 2 electrons are required and 1 hydrogen ion are required to produce NADPH. Okay, ingat 2 elektron. Saya tulis baik semula lah, cantik sikit. NADP plus 2 electrons plus H plus to form 1 molecule of NADPH. Okay, hydrogen ion comes from the stroma, electrons comes from the ETC. Okay, and NADPH is released into the stroma because Calvin cycle occur in the stroma to produce sugar. Okay, nampak release into the stroma. So point number 4, NADP plus accept 2 electrons, 1 protons to reduce to NADPH. So NADP plus as the terminal electron acceptor. Ingat NADP plus as the terminal electron acceptor, and NADPH is released into the stroma. Kalau cellular respiration, the final electron acceptor will be oxygen molecule. Itu beza dia. Okay. Alright, so ini kita balik semula kepada photosystem 2. tadi kan. So tadi photosystem tu dia punya P680 dah release electron. And so dia jadi oxidized. It is missing an electron. Macam mana nak ganti the missing electron so that from P680 dia jadi P680 balik semula? Kan? To reduce. Okay, to reduce P680 plus to become P680 whole kan. So cara nak gantikan elektron tu adalah melalui proses photolysis of water. Okay, photolysis of water, splitting of water using light energy. Okay, so P680 becomes P680 plus. Okay, missing an electron. It is oxidized. When it loses an energized electron to the primary electron acceptor. P680 plus, okay, is a strong oxidizing agent. Okay, it is a strong oxidizing agent. So it pulls electron away from from an oxygen atom of a water molecule. Okay, from an oxygen atom of water molecule. So water molecule is split by an enzyme into 2 electrons, 2 hydrogen ions and 1 oxygen atom. Okay, so the electron are transferred one by one to P680 plus, thus reducing it to P680. Okay, so the hydrogen ions are released into the thylakoid lumen. So the two water molecule are required, okay, to be split to form one molecule of oxygen, okay, which is released as byproducts. So kat sini dia punya apa equation dia, one water molecule will be released to form 2 hydrogen ion plus 2 electron plus 1 oxygen atom. So to produce one molecule of oxygen, you require 2 water molecule to be split, okay, to release one oxygen molecule. Ingat so oxygen molecule is the byproduct of light reaction. Okay, light reaction yang berlaku dekat photosystem tu, sebab photosystem tu akan berlaku photolysis of water. Okay, so this is the whole diagram lah, kan, gabung-gabung semua yang tadi, so dia dapat yang ni lah. So daripada gambar ni awak boleh sepatutnya cuba balik nanti cuba tengok balik gambar ni, cuba explainkan macam mana light reaction berlaku. Okay, macam mana light reaction berlaku yang melibatkan non-cyclic electron transport. So ingat dia berlaku start daripada photosystem 2 and then reaches photosystem 1. Okay, and then the electron will be released and accepted by an ADP plus, and last kali dia akan diterima oleh an ADPH molecule. Okay, so this is released into the stroma. Okay, kalau awak tengok kat note sini, kat slide ni, the hydrogen ion, which is the product of splitting of water molecule, is released into the thylakoid lumen. Okay, ingat kalau ini thylakoid, right? Thylakoid membrane. Dalam ni adalah thylakoid lumen. Okay, so photosystem ni it is embedded in the thylakoid membrane. Alright, so the splitting of water dia akan berlaku kat dalam thylakoid. Dia akan berlaku kat dalam thylakoid dan menyebabkan hydrogen ion ni accumulates in the thylakoid lumen. Okay, and causes hydrogen ion concentration in the thylakoid lumen to be high. Okay, concentration of hydrogen ion in the thylakoid lumen is high due to the splitting of water molecule. Kalau kita tengok je ya gambar ni, nampak? So the gambar ni dia nak menunjukkan splitting of water, it occurs in the thylakoid space. Thylakoid space sama juga dengan thylakoid lumen ya. Okay. Cuma NADPH tu akan release dekat stroma. Same goes to ATP. It is produced and released in the stroma sebab Kevin cycle berlaku dekat dalam stroma. So concentration of hydrogen ion in the lumen is really high compared to concentration of hydrogen ion in the stroma is low. Okay. Okay, so nanti awak tengok balik gambar ni, cuba extendkan daripada awal yang berlaku start daripada photosystem 2 sampai dekat photosystem 1. Okay, photosystem 1. Okay, so ingat ya, photosystem 2 dia ada P680 akan release elektron dia jadi P680 plus oxidized form. P700, okay, dia akan dalam bentuk oxidized receive elektron daripada photosystem 2, okay, so dia akan jadi P700. So absorb energy balik photon, kan, so dia akan excite and release dia punya electrons, okay, and it is sampailah dia akan hasilkan NADPH, okay. So for non-cyclic electron transport, in the presence of light, the non-cyclic electron transport involve the formation of ATP, okay, which is coupled by one-way flow of energized electron from water, okay, until it is received by NADPH molecule. Ni salah NADPH, bukan NAPDH NADPH, betul kan? Okay, so the product of light reaction ATP and NADPH and betul kan NADPH, alright, are released into the stroma and are required by the carbon fixation reaction of the Kelvin cycle. Okay, Kelvin cycle produce sugar. Banyak kali dah cakap ni. Kelvin cycle produce sugar, it occurs in the stroma. Okay, tadi non-cyclic. Nama pun non-cyclic one way flow. Mula elektron tu daripada water, received by photosystem 2, pergi along transport electron transport chain, received by photosystem 1, and then received last kali by NADPH. Okay. So elektron tu dia tak akan dapat semula. Dia akan diambil oleh NADPH. Tapi kalau untuk cyclic electron flow, okay, only involve photosystem 1, it is a cyclic pathway, okay. So cyclic tu, kenapa cyclic? Dia melibatkan pergerakan elektron lah, kan. So macam saya cakap tadi, dia boleh juga dipanggil sebagai cyclic photophosphorylation. Photophosphorylation. Okay. Dia perlukan energy in the form of light energy untuk menghasilkan ATP. Okay. So cyclic hanya akan hasilkan ATP sahaja. So the P700 absorb light energy and excite its electron and are transported through an electron transport chain and eventually return back to P700. So it is a cyclic process. Okay. From P700, okay, absorb photons, excited, excite the electron release, received by primary electron acceptor, FDD received by paradoxine, okay. Paradoxine, and then received by cytochrome complex, and then by PC. PC is plastocyanin. Plastocyanin and back to P700. Tadi saya mentionkan PQ kan? PQ adalah plastoquinone. So dia hanya terlibat untuk non-cyclic. Okay? Non-cyclic. So kalau electron carrier untuk non-cyclic, dia daripada primary electron acceptor, PQ, plastoquinone, cytochrome complex, PC, plastocyanin sampai dia reach P700 and to paradoxine and to NADP plus reductase, okay, which is the enzyme to catalyze the production of NADPH. Itu non-cyclic. Kan non-cyclic yang ni. Okay, yang ni. Tapi kalau cyclic, yang ni. Okay. Daripada P700 back to P700. Okay. So the energy release is used for the synthesis of ATP. Kan? So as the electron travels, it releases energy to synthesize ATP during chemiosmosis. Okay, chemiosmosis. So tak ada penghasilan NADPH, tak ada melibatkan oxygen release. Okay, sebab dia hanya melibatkan photosystem 1. Photosystem 1. Photosystem 1 tak ada berlaku photolysis of water. So maksudnya tak akan ada release of oxygen molecule. Okay, so significance of cyclic electron transport. Ini adalah antara soalan yang suka keluar dalam exam. Apa pentingnya cyclic electron transport ni? To generate extra ATP. Ataupun kalau baca dalam buku Campbell, digunakan surplus. Okay, excess ATP, extra ATP, surplus ATP, sama je. To power other ATP requiring process in the chloroplast and help maintain the optimal ratio of ATP to NADPH required for carbon fixation and Calvin cycle. Kalau kita tengok Calvin cycle nanti, Calvin cycle akan menggunakan lebih banyak ATP molecule. Okay, Calvin cycle akan menggunakan lebih banyak. It requires much more ATP molecule compared to an NADPH molecule. So this will generate the extra ATP that is needed during Calvin cycle. Okay. Jadi kita tengok ada dia punya fix number, berapa banyak ATP yang diperlukan, berapa banyak NADPH diperlukan yang awak kena ingat. Okay. So this table shows the comparison of linear and cyclic electron transport. Okay. So kalau linear tu maksudnya non-cyclic lah, ingat eh. Linear juga dikenali sebagai non-cyclic. So boleh guna mana-mana. Non-cyclic ataupun linear sama. Linear sebab apa? Daripada air ke PS. Okay, daripada air, elektron tu daripada air ke PS2 along ETC. Okay, until received by PS1 and then ferrodoxin dapat FDN. Sampai diperolehi oleh NADPH. So it is a linear flow of electron. Okay, non-cyclic. Dia tak balik semula ke PS2. Okay. Tapi kalau cyclic electron transport, it is electron source none, sebab dia memang elektron kepunyaan P700. Okay. So dia akan travel daripada P700 balik semula ke P700 of photosystem. Photosystem 1. Okay. Daripada P700 balik semula ke P700. So linear electron flow or non-cyclic is oxygen release? Yes. Sebab apa? Berlaku photolysis of water. Kalau cyclic is oxygen release? No lah, sebab no photolysis of water. Okay. So kalau linear electron transport, terminal electron acceptor adalah NADP plus before it becomes NADPH, okay, which is the product of light reaction requires for Calvin cycle. So terminal electron acceptor tak ada sebab dia adalah cyclic process. Daripada P700 balik semula P700. It is a cyclic process. So maksudnya tak ada penghujung lah. Kan tak ada awal, tak ada penghujung. Okay, so kalau linear form in which energy is temporarily captured. Okay, so energy daripada light, okay, will be used to synthesize ATP and also NADPH. So these are basically daripada light energy jadi chemical energy. Okay, chemical energy stored in the form of chemical energy, which is the production of ATP and also NADPH. So energy is temporarily captured and stored in the form of ATP. Okay, so both production of ATP is through chemiosmosis. And then for photosystem ni, kalau linear, requires both photosystem 2 and also photosystem 1. Kalau cyclic, photosystem 1 only. Okay. Okay, so yang slide seterusnya ni nak explain pasal chemiosmosis lah kan. So ini kena fahamkan perbezaan concentration hydrogen ion dalam thylakoid lumen dan juga stroma. Ingat lumen will have high concentration of hydrogen ion, stroma will have low concentration of hydrogen ion. Okay, so as electron moves down ETC, energy is released and used to pump. Kenapa pump? Dia adalah jenis active transport. Okay, pump protons, which is hydrogen ion, from stroma. Nampak stroma concentration of hydrogen ion is low, into thylakoid lumen, which is high concentration of hydrogen ion. Okay, dia pergerakan daripada low concentration to high concentration of hydrogen ion. So it is a form of active transport. Active transport tahu dia perlukan energy, dan energy datangnya daripada electron. Okay, electron that travels along ETC. Okay, so forming a proton gradient across the thylakoid membrane. Proton gradient ni nanti akan menghasilkan sejenis tenaga, we call it as proton motive force. Okay, proton motive force. So this proton motive force, okay, will cause hydrogen ion to diffuse from the lumen into the stroma. Sebab apa? Concentration of hydrogen ion dalam stroma dah tinggi. Awak tengok kat sini kan, dah tinggi. So when there is a high concentration of hydrogen ion in the lumen is high, so they can diffuse back into the stroma. Okay, so this is due to the proton motive force, okay, that causes ATP synthase, okay, to synthesize ATP from ADP. So for proses ini dikenali sebagai photophosphorylation. Photo sebab apa? Menyebabkan light energy, okay, and that is used to synthesize ATP, okay. ATP synthase catalyzes the photophosphorylation of ADP to ATP. Okay, so ATP is actually produced in the stroma. Okay, so kalau awak tengok kat gambar Rajah ni, okay, awak tengok this is the structure of ATP synthase. Ini saya bulatkan ni. Dia punya catalytic knob ni is facing the stroma. Okay. So when the catalytic knob is facing the stroma, obviously the production of ATP occurs in the stroma. Cuma pergerakan ion tu kan, when there is a high concentration of hydrogen ion in the lumen, the hydrogen ion will diffuse down, okay, through the ATP synthase and catalyzes ATP. Okay, so sama. Ini gambar rasa yang sama, cuma dia dalam bentuk 3D lah kan. So dia dalam bentuk 3D. So bila 3D orang nampak, okay, kat sini ada thylakoid membrane kan, the yellow hydrophobic tail of phospholipid kan. So these are thylakoid membranes. Okay, thylakoid membranes. So in the thylakoid membranes you have photosystem 2 embedded kan. This is photosystem 2 kan. You have also photosystem 1. Okay, and also component of electron transport chain. You have plastoquinone, cytochrome complex, plastocyanin, and also ferrodoxin. Okay, so these are component of ETC. They are electron carriers of ETC. Okay, so point number 1, electron travels along ETC. So the electron carriers within the membrane, within the thylakoid membrane, becomes alternately reduced and oxidized. Reduce maksudnya apa? Receive electron and then donate balik. Receive electron and then donate balik. Receive electron and then donate balik. So it transfers electron. Okay, they accept and donate electrons. Okay, so energy released during electron transport is used to pump hydrogen ion. Pump is active transport from the stroma into thylakoid lumen. Stroma, low concentration of hydrogen ion. Thylakoid lumen, high concentration of hydrogen ion. So it is a type of active transport. Okay, so thylakoid lumen has high concentration of hydrogen ion that accumulates in the thylakoid lumen. So the hydrogen ion also results from the splitting of water molecule. So sekarang ni ada dua sumber, okay, two source of hydrogen ion in the thylakoid lumen. Satu from splitting of water molecule at photosystem 2, kan? Satu lagi result from the proton pump. Okay, that pump hydrogen ion from stroma into the thylakoid lumen. Kan? So that's why concentration of hydrogen ion in the lumen is really high. So when there is a high concentration of hydrogen ion in the lumen, so this creates a proton gradient across the thylakoid membrane, which is known as the proton motive force. Okay, so when there is a high concentration of hydrogen ion in the lumen, so it causes hydrogen ion to diffuse back. Okay, hydrogen ion diffuse back into the stroma through the ATP synthase, okay, in the thylakoid membrane. So the hydrogen flows through ATP synthase, generate ATP. Okay, generate ATP. So basically ATP synthase will use a form of energy. We call it as the proton motive force. Pergerakan hydrogen ion tu daripada high concentration to low concentration creates proton motive force, so that the energy is used by ATP synthase to generate ATP. Okay, so kalau tengok kat gambar ni, 4 hydrogen ion are required by the ATP synthase to synthesize 1 molecule of ATP. Okay, every 4 is used to synthesize 1 molecule of ATP, every 4 hydrogen ion. Okay, so dekat last table ni kan, it shows you a comparison of chemiosmosis in chloroplast and also mitochondria. Ingat chemiosmosis berlaku dalam kedua-dua proses cellular respiration dan juga photosynthesis. Okay, which is to be specific during light reaction. Okay, Kevin cycle tak melibatkan chemiosmosis. Okay, so both organelles generate ATP by chemiosmosis. Ini adalah similarity. Soalan ni adalah boleh dia antara soalan yang boleh keluar lah kan. You have to compare between chemiosmosis in chloroplast and also mitochondria. Contoh soalan lah. Okay, so dia punya differences. For chloroplast, light energy into the chemical energy of ATP. So chloroplast will use light energy and convert the energy into chemical energy in the form of ATP. But for mitochondria, it is from chemical energy. Cellular respiration requires apa? Glucose kan? Glucose. Okay, glucose from food. Okay, so glucose will be broken down. Ingat lagi proses glycolysis and then link reaction and then Krebs cycle, baru masuk oxidative phosphorylation, baru dia akan menghasilkan ATP. Okay, ingat balik proses cellular respiration. Okay, so daripada glucose sampai oxidative phosphorylation baru akan menghasilkan ATP melalui proses chemiosmosis. Kalau another difference, okay, pergerakan hydrogen ion tu. Okay, kalau dalam chloroplast, so the hydrogen ion pumped into thylakoid space from stroma. Okay, from stroma into thylakoid space. Okay, low concentration of hydrogen ion in the stroma to thylakoid space, which has high concentration of hydrogen ion, and then back hydrogen ion diffuse back into the stroma. Bila dah tinggi sangat dalam thylakoid space, so dia akan diffuse back into the stroma through ATP synthase. And ATP synthase produce ATP in the stroma. Okay, tapi kalau mitochondria, dia punya pergerakan hydrogen ion tu daripada mana? Daripada intermembrane space back into, pump, okay, pump from matrix into intermembrane space. Matrix have low concentration of hydrogen ion, intermembrane space have high concentration of hydrogen ion. Ingat balik cellular respiration, pump maksud dia apa? Active transport. Okay, hydrogen ion is pumped from the matrix into into intermembrane space. Mana nak datang energy from pump tu? From electron transfer lah along ETC. Okay, ETC dekat mana? Dekat crystal. Ingat balik proses cellular respiration ya. So once hydrogen ion is high in the intermembrane space, so dia akan diffuse back into the matrix through ATP synthase. Okay, ATP synthase. So ATP synthase dia punya catalytic knob dia terletak dalam matrix. Okay, so the production of ATP is in the matrix. Okay. So inilah ini gambar raja digabungkan kedua-dua chemiosmosis untuk menunjukkan perbezaan chemiosmosis dalam mitochondria dan juga dalam chloroplast. Yang the darker region here ni, it shows you there is a high concentration of hydrogen ion. The lighter region here, it shows you the low concentration of hydrogen ion. Okay, so kalau belah sini, okay, sebelah sini menggambarkan mitochondrial, sama je. It's just you have to know the region. Okay, kalau mitochondria melibatkan apa? Region apa? Matrix dengan intermembrane space. Matrix, low concentration of hydrogen ion. Intermembrane space, high concentration of hydrogen ion. So macam mana nak gerakkan dia? Melalui energy released by electrons as the electron travels along ETC. Okay, so as electron travels along ETC, energy is released and the energy is used to pump hydrogen ion from matrix into intermembrane space, and then when there is a high concentration, so they can diffuse back through ATP synthase back into matrix, okay, and produces ATP. Sama juga kalau masa dalam chloroplast kan, so dia akan pump daripada stroma, okay, which has low concentration of hydrogen ion, into the thylakoid space because there is a high concentration of hydrogen ion in the thylakoid space. Okay, so dia akan diffuse back balik ke dalam stroma dan proses tu akan menghasilkan ATP. Okay, dia menggunakan apa tadi? ATP synthase akan menggunakan tenaga apa? Proton motive force. Okay, high concentration of hydrogen ion diffuse back into the low concentrated hydrogen ion region, okay, through ATP synthase. Okay, kita break sekejap. Okay, keluarlah dulu. Okay, thank you.
Thank you, madam.
Okay.
Thank you, madam.
Thank you, madam.
Thank you, madam.
Thank you, madam.
Okay.
Madam, nanti yang madam ikut ni, madam pun dekat channel madam tu.
Nanti saya
thank you,
letak
madam.
dekat saya punya channel. Okay. Sorry, saya terkeluar lagi. Sekejap ya, saya nak cuba apa? Connect. Sekejap ya, saya nak connect. Saya nak tethering jap. Kalau saya terkeluar, bagi tahu WhatsApp saya.
Okey, madam.
Okay. So sekarang ni kita nak tengok last part. Tak ada last part. Bahagian-bahagian akhir which is the second part of okay. So dark reaction kita nak panggil dia sebenarnya adalah sebagai Kevin cycle. Okay. So walaupun nama dia sebagai dark reaction, dia bukannya berlaku pada
Madam. Sangkut madam, kami tak dengar.
Okay, sorry once again. Alright. So tadi saya explain sampai awal-awal, baru awal lagi ke?
Ya madam, dekat dark reaction.
Okay, saya explain balik semula ya. Kelvin cycle dia juga dikenali. Saya explain tak kenapa Kelvin cycle? Ha.
Tak, madam tak present.
Oh ya. Dah. Okay, sekejap eh. Okay. Tadi saya explain tak kenapa Kelvin cycle tu dinamakan sebagai dark reaction?
Tak sempat madam.
Tak sempat. Okay. Kita start dari semula ya untuk dark reaction. Okay. Okay. Dark reaction ni juga dikenali sebagai Kelvin cycle. Okay. Walaupun dia dinamakan sebagai dark reaction, awak kena ingat dia Kelvin cycle ni perlu berlaku pada waktu siang. Sebab apa? Kelvin cycle dia perlukan products of light reaction. Kelvin cycle requires ATP and also NADPH which is produced during daylight. Okay. Sebab apa daylight? Sebab daylight diperlukan oleh light reaction untuk hasilkan ATP dan juga NADPH. So, walaupun Kelvin cycle ni kita refer juga dia sebagai dark reaction, tapi sebenarnya Kelvin cycle perlu berlaku pada waktu siang. Kelvin cycle will require light energy indirectly. Okay, so it means that Kelvin cycle has to occur during daylight also, walaupun nama dia dikenali sebagai dark reaction. Dark reaction sebab Kelvin cycle tak memerlukan light energy secara directly. Faham ke? Boleh?
Faham madam.
Okay. So jangan ingat Kelvin cycle ni kita menamakan dia sebagai dark reaction, dia boleh berlaku pada waktu malam. Tak. Kelvin cycle hanya akan berlaku pada waktu siang sebab dia perlukan NADPH dan juga ATP yang terhasil semasa light reaction. Okay. So Kelvin cycle occurs in the stroma sebab apa? ATP dengan NADPH terhasil dalam stroma. Okay. Dan juga enzyme that is involved in Kelvin cycle are located in the stroma. So Kelvin cycle ni kalau tengok tajuk dia bagi nama dia sebagai carbon fixation. Okay, carbon fixation. Kenapa carbon fixation? Kalau tengok fixation maksudnya incorporate. Okay, so for Kelvin cycle, the Kelvin cycle will incorporate carbon dioxide to another organic molecule. We call it as carbon dioxide acceptor. Okay, so for Kelvin cycle, the molecule that function as carbon dioxide acceptor is known as RUBP, ribulose biphosphate. Okay, so carbon enters the cycle as carbon dioxide and leave as a sugar named glyceraldehyde triphosphate, which is a 3-carbon sugar. Okay, so Kelvin cycle is also known as C3 pathway because the product of initial carbon fixation is a 3-carbon compound. Okay, plant initially fix carbon, this way is called as C3 plant. Carbon dioxide, okay, is a 1-carbon compound. Okay, bila digabung dengan RUBP is a 5-carbon compound. Okay, so dia punya produk dia akan hasilkan a molecule which is a 6-carbon compound that is really unstable. Okay, it is unstable. So this carbon compound akan split into 2 molecule of 3-carbon compound. 2 molecule of 3-carbon compound yang dikenali sebagai PGA. Okay, PGA. PGA is a 3-carbon sugar. So that's why kita namakan dia sebagai C3 pathway. The product of the initial carbon fixation is a 3-carbon sugar which is PGA, phosphoenol pyruvate. Bukan? Phosphoglycerate. Okay, phosphoglycerate. phosphoglycerate. Okay, ataupun boleh letak 3 kat depan. 3-phosphoglycerate. Okay, sebab phosphate lekat pada karbon nombor 3. Okay, so these plants are known as C3 plants. Okay, so carbon cycle is anabolic. Okay, anabolic maksud dia apa? Maksud dia require energy. Okay, so the energy comes from ATP and also NADPH function as reducing agent. Okay, it's used in the formation of organic molecule which is sugar from carbon dioxide. So carbon cycle it has 3 phases. The first phase is carbon dioxide uptake ataupun kita boleh panggil dia sebagai carbon fixation phase. Boleh nama mana? Bergantung pada buku yang awak refer. Kalau Campbell dia panggil sebagai carbon fixation. Kalau buku Solomon yang awak beli tu dia panggil sebagai carbon uptake phase. Okay. So phase yang kedua adalah carbon reduction phase. Phase yang ketiga adalah RUBP regeneration phase. Ingat RUBP function as carbon dioxide acceptor. Okay. So alright. So the first phase, carbon dioxide uptake ataupun carbon fixation phase. Okay. So carbon dioxide reacts with RUBP. RUBP is a 5 carbon compound. Dia bertindak sebagai carbon dioxide acceptor. Okay, acceptor. So the reaction that combines carbon dioxide dengan RUBP is catalyzed by the enzyme ribulose biphosphate carboxylase ataupun ribulose biphosphate oxygenase ataupun Rubisco. Okay, nama pendek dia adalah Rubisco. Okay, so nanti kita tengok kenapa nama dia ada boleh oxygenase. Okay, so the reaction produce one molecule of unstable 6 carbon intermediate that breaks immediately into 2 molecule of 3 carbon compound which is PGA. Okay, so the carbon of the carbon dioxide molecule has been fixed or incorporated into a pre-existing carbon skeleton. Pre-existing carbon skeleton maksudnya dalam cycle tu dia bergabung dengan RUBP lah. Okay, alright, so it fixed ataupun incorporate carbon dioxide campur dengan RUBP, produce a 6 carbon intermediate that breaks down into PGA, 2 molecule of PGA. So kalau tengok kat dalam gambar ni, okay, so 1 molecule of glucose to produce 1 molecule of glucose which is a 6 carbon sugar. Okay, the carbon cycle requires 6 molecule of carbon dioxide. Ingat matlamat carbon cycle untuk hasilkan sugar. Okay, sugar yang terhasil masa photosynthesis adalah glucose. Okay, glucose is a 6 carbon compound. Okay, so untuk menghasilkan glucose which is a 6 carbon compound perlukan 6 molecule of carbon dioxide. Okay, so 6 molecule of carbon dioxide will combine with 6 molecule of RUBP and the enzyme is catalyzed and the reaction is catalyzed by the enzyme Rubisco ribulose biphosphate carboxylase and produce a 6 carbon intermediate which is unstable. 6 molecule unstable 6 carbon intermediate. Tengok gambar kat sini Ini tak salah saya nak draw sampai 6 kali kan. So maksudnya kat sini satu carbon campur dengan 1 RUBP. Okay. Akan hasilkan 1 molecule of 6 carbon intermediate. Tapi sebab nak hasilkan glucose, so awak kena imagine cycle tu berlaku 6 kali serentak. Okay, 6 kali serentak. So maksudnya semuanya awak kena kalikan dengan 6. Tapi bila dah hasilkan 6 carbon intermediate ni, dia akan split jadi 2. Kan? So bila dia split jadi 2, so produk yang terhasil adalah phosphoglycerate 12 molekul. 6 tadi dengan 2 sebab dia split jadi 2. So you will produce 4 molecule of phosphoglycerate. Okay. So kenapa the 6 carbon intermediate ni dia split? Sebab molekul tu ada 2 phosphate. Kita belajar molekul yang ada 2 ada phosphate group yang get attached to it is a highly energized molecule. highly energized molecule. So because they're highly energized, it is really unstable. Okay, unstable. So they akan split. Okay, they akan split. So 6 carbon split jadi 2 each. So you produce 12 molecule of phosphoglycerate, which is PGA. So this is the first product of carbon fixation. Carbon fixation, which is a 3 carbon compound. Sebab tu kita namakan sebagai C3 pathway. Okay, C3 pathway. The first product of first carbon fixation is a 3 carbon compound, which is PGA. Okay, so untuk phase yang kedua sekarang ni adalah reduction phase. Okay, so when enters into the reduction phase, the PGA molecule, 12 molecule of PGA is phosphorylated by 12 molecule of ATP. Okay, so PGA ni dia ada satu fosfat kan. Bila ATP masuk, ATP akan bagi dia punya fosfat, ATP akan jadi ADP. Okay, so dia akan release terminal fosfat dia. So dia akan attach kat molekul PGA ni. So dia akan jadi a molecule, we call it as ni molekul ni kita namakan sebagai 1,3 bis phosphoglycerate. Okay, 1,3 bis phosphoglycerate. Okay, 1,3 bis phosphoglycerate. So this 1,3 molecule bis phosphoglycerate will be reduced by 12 molecule of NADPH. Okay, when it is reduced, so dia akan jadi dia akan buang fosfat dia akan jadi 12 molecule of glyceraldehyde 3 phosphate. Nampak? Remove phosphate dia akan jadi 12 molecule of G3P. Dan ingat proses tu memerlukan 12 molecule of NADPH to reduce this to this one. Okay, to reduce 1,3 bis phosphoglycerate to become glyceraldehyde 3 phosphate. Okay, daripada 12 molekul G3P yang terhasil semasa phase yang kedua ni, 2 G3P akan leave the cycle, 2 molecule of G3P will leave the cycle to form 1 molecule of glucose. Kenapa 2 G3P? Sebab satu G3P is a 3 carbon compound. So perlukan 2 untuk hasilkan 1 molecule of glucose. Okay, so 12, 2 keluar, so 10. Okay, 10 remain in the cycle. So the 10 G3P that remains in the cycle will enter into the third phase, which is RUBP regeneration phase. Okay, so 10 G3P molecule, which is it will total up to 30 carbon atoms. 10 kali dengan 3 carbon compound kan. So total dalam cycle tu sekarang ni kita ada 30 carbon atom in the cycle. Okay, to regenerate back 6 molecule of RUBP. RUBP ingat is a 5 carbon compound. Okay, so 5 carbon compound kali dengan 6 molecule, total berapa? 30 carbon in the cycle. So maksudnya kenapa 10 GTP kena remain? Sebab hasilkan 6 molecule of RUBP. Okay, 6 molecule will be 6 molecule of RUBP will be used to synthesize, okay, to be synthesized. Okay, tapi kat sini dia tak adalah nak menghasilkan RUBP lagi. Sebab apa? Kat sini bila 10 G3P remain in the cycle, dia akan undergo rearrangement first. Okay, saya terskip tadi. Rearrangement first, rearrange into 6 molecule of ribulose phosphate. Ribulose phosphate is still a 5 carbon sugar. Okay, so from the 6 ribulose phosphate, it is phosphorylated by 6 molecule of ATP, baru boleh terhasil 6 molecule of RUBP, ribulose biphosphate. Ada dua phosphate attached to it. Okay, RUBP is a 5 carbon compound. 6 molecule kali dengan 6 sama dengan total 30 carbon in the cycle. sama kan? So 30 at the beginning of the phase. Okay. So RUBP is regenerated and the new cycle begins. Okay. So kalau tengok ke gambar ni, so ingat matlamat Calvin cycle untuk hasilkan sugar. Okay, matlamat photosynthesis untuk menghasilkan glucose. One molecule of glucose is a 6 carbon compound. So kita perlukan 6 carbon dioxide to enter. Okay. First phase is carbon dioxide uptake phase. Okay. And then dia akan hasilkan berapa? 12 molecule of PGA. Okay. So 12 molecule of PGA akan mengalami phosphorylation, akan reduced, hasilkan 12 molecule of G3P. Okay. So daripada 12 G3P yang terhasil, 2 G3P akan exit the cycle to form glucose and other carbohydrates. Okay. So another 10 remain in the cycle to enter into the RUBP regeneration phase. So during regeneration phase, untuk menghasilkan 6 RUBP perlukan 6 ATP. Okay, so during RUBP regeneration phase requires 6 molecule of ATP to synthesize 6 molecule of RUBP. So kalau tengok tadi, ingat tadi balik cyclic electron flow kan? Cyclic electron flow to produce excess or extra ATP kan? Kalau tengok dekat Calvin cycle ni, berapa ATP is for one glucose kan? For one glucose molecule, berapa ATP yang diperlukan? 12 masa phase 2 dengan 6 masa phase 3. So berapa altogether? For Calvin cycle required to produce one glucose, it is 18 molecule of ATP. Okay, berbanding dengan NADPH, berapa yang dia require? 12 sahaja. Sebab dia hanya akan digunakan masa reduction phase. Faham? So that is another significance of cyclic electron flow to produce excess or extra ATP that is required during the Calvin cycle to balance the ratio kan, for the usage of ATP and NADPH during Calvin cycle. Okay. Okay, so summary of the Calvin cycle requires the product of light dependent reaction. So walaupun dia nama dark reaction, tapi dia perlu berlaku during daylight. Okay, during daylight. So every 6 molecule of carbon dioxide enters the cycle. 6 carbon leave the cycle as 2 molecule of G3P, sebab 2 molekul ni akan hasilkan 1 molekul glucose. Okay, so 2 molecules of G3P produce 1 molecule of glucose or fructose. Okay, so plant uses glucose to produce starch and also cellulose. Cellulose is the building block of cell wall for plant cell. Okay, so glucose and fructose are joined to produce sucrose. Sucrose can be harvested from sugar cane, sugar beet and also maple sap. Okay, maple syrup lah. Alright. So kalau makan buah dia manis semua tu disebabkan oleh penghasilan fructose or glucose during Calvin cycle of photosynthesis. Okay, so kita masuk sikit lagi part last. Larat lagi ke? Kelas, larat lagi?
InsyaAllah boleh, Madam.
Boleh, Madam.
Okay, habiskan sikit lagi Okay, so C3, C4 and Chemplan. Saya summarize lah. Okay, so photosynthesis untuk C3, C4 dengan Chemplan. C3 ni adalah kumpulan tumbuhan yang paling banyak. Okay, yang paling banyak ada. So dia akan undergo the normal Calvin cycle to produce sugar. Tapi untuk Chemplan, dia adalah tumbuhan yang tumbuh dekat tempat yang panas. Tempat yang panas. Okay. Hot and arid area kan. So bila dia tumbuh kat tempat yang panas, dia untuk mengurangkan kehilangan air melalui transpiration. Okay. Dia akan dia akan close. Okay. Close stomata. close stomata to reduce water loss. Reduce water loss during hot dry day. Okay. So, but the consequence of closing stomata, carbon dioxide cannot enter into the leaf. When carbon dioxide cannot enter into leaf, Calvin cycle cannot occur. Okay. When cycle cannot occur, sugar cannot be produced. Okay. So, Chem and C4 plant.
Suara tak jelas.
Jelas tak? Hello?
Tak ada.
Putus-putus.
Aku tengah ya.
Sekejap ya.
Sekarang okey tak?
Okey lah.
Okey, dengar? Jelas?
Jelas ada. Jelas.
Okey. So kalau untuk C4 dengan chem plant, dia adalah tumbuhan yang duduk kat tempat yang panas. Okey. So bila dia duduk tempat yang panas, untuk mengurangkan kehilangan air, okey. So dia akan close stomata. So bila stomata closed, consequences dia adalah carbon dioxide cannot enter into the leaf, and bila carbon dioxide cannot enter the leaf, carbon cycle cannot occur and sugar cannot be produced. Okay. So ini akan lead kepada photorespiration. Kan, dia akan lead kepada photorespiration. Okay. Photorespiration yang berlaku, dia berlaku akibat daripada carbon dioxide concentration in the leaf is low. Okay. Dia low sebab apa? Stomata closed. Tak ada carbon dioxide yang boleh masuk dalam leaf, ataupun stomata ni akan close partially. For C4 plant, stomata akan close partially. Okay, maksudnya still ada carbon dioxide boleh masuk tapi sikit. So bila carbon dioxide concentration is low, kedua-dua plant ni perlu ada mechanism of adaptation. Okay, dia perlu ada mechanism of adaptation untuk mengatasi masalah photorespiration ni. Okay, bila photorespiration ni berlaku, bila carbon dioxide concentration low, dan enzim dalam Calvin cycle ni, apa tadi enzim? Rubisco kan? Rubisco ni, dia punya kekurangan ataupun lacking dia adalah dia tak boleh bind kepada low concentration of carbon dioxide. Rubisco ni dia adalah enzim yang lagi prefer kepada oxygen. Okay, so Rubisco will bind to oxygen when oxygen concentration in the leaf is high compared to carbon dioxide. Kenapa bila stomata ni tutup, okay, oxygen concentration is high? Sebab oxygen is the byproduct of light reaction. Ingat tadi kan? Light reaction, dia punya byproduct adalah oxygen. Tapi bila stomata tutup, macam mana oxygen nak keluar? Oxygen akan accumulate dalam leaf. So bila oxygen accumulate dalam leaf, Rubisco ni dia tak nak bind kepada carbon dioxide, dia bind kepada oxygen. Okay, so bila Rubisco bind kepada oxygen, dia akan menghasilkan satu compound, nama dia glycolates, dan compound tu akan di metabolizkan, glycolate. Okay, so compound ni akan di metabolizkan dekat mitochondria, dan mitochondria akan instead of proses to hasilkan ATP, dia akan use ATP. ATP is used. Sekejap. There's no ATP produced, dan juga dia akan menghasilkan carbon dioxide. Okay, so photorespiration ni dia akan mengurangkan photosynthetic yield. Sebab apa? Carbon dioxide tak boleh fit into Calvin cycle and sugar cannot be produced. Itu adalah photorespiration. So for chem dengan C4 plant ni, dia kena atasi masalah photorespiration ni, dia kena berlaku adaptation. Okay. Macam mana nak berlaku adaptation? fixation, dia akan melakukan carbon fixation twice. Okay, C4 dengan Chemplan ni dia akan lakukan carbon fixation twice. Okay, so the first carbon fixation is done by the first carbon dioxide acceptor, which is PEP, phosphoenopyruvate, and the second carbon fixation adalah melalui proses Kevin cycle yang biasa, which is by RUBP. Adaptation by C4 dengan Chem ni, dia akan ada satu lagi enzim, which is kita namakan dia sebagai PEP carboxylase. Okay, PEP carboxylase, dia punya ability adalah dia boleh combine dengan carbon dioxide instead of oxygen. Tadi kita tengok kan carbon dioxide concentration is low. By having PEP carboxylase will bind to carbon dioxide and dia tak combine langsung kepada oxygen. Okay, so that is the first fixation, carbon fixation. Sebab dia boleh adapt, sebab dia ada enzim PEP carboxylase. Okay, hanya akan bind kepada carbon dioxide walaupun carbon dioxide concentration in the leaf is low. Okay. Okay, so itu photorespiration lah saya dah explain kan. So photorespiration berlaku pada hot dry day disebabkan oleh mata close to conserve water. So this limit photosynthesis ataupun reduce photosynthetic yield, reduce the production of sugar. Okay. So the closing of stomata will reduce carbon dioxide that can enter into the leaf and causes oxygen concentration to increase sebab dia adalah byproduct of light reaction. Okay, so that's condition favors a seemingly wasteful process which is photorespiration. So kalau tengok gambar ni, okay, during hot dry day, stomata close kan, so carbon dioxide tak boleh masuk, oxygen tak boleh keluar. Dia punya benefit dia hanya adalah pada dia akan reduce water loss, reduce transpiration, tapi dia akan menyebabkan photorespiration berlaku and reduce photosynthetic yield. Okay. Okay, so tadi saya cakap contoh kebanyakan plant adalah C3 plant. Contoh C3 plant, soybean, potato, rice, wheat, oats, kebanyakan tumbuhan adalah C3 plant. Ini kan soalan ni pernah keluar dalam exam. Dia suruh bagi contoh C3 plants. Okay, bagi yang ni je. Alright, jangan nak bagi yang lain-lain. Okay, so initial fixation of carbon dioxide is via Rubisco. Okay, to form a 3 carbon compound which is phosphoglycerate. Ini yang normal Kevin cycle. Okay, normal Kevin cycle dia akan punya Rubisco dan hasilkan phosphoglycerate which is a 3 carbon compound. Tapi bila photorespiration berlaku disebabkan oleh carbon dioxide concentration is low, okay, Rubisco akan fix oxygen. Dia akan prefer oxygen. So sebab tu nama Rubisco ni tadi ribulose biphosphate carboxylase ataupun oxygenase. Okay, sebab Rubisco ni dia akan prefer oxygen berbanding dengan carbon dioxide. Okay. So the product splits and 2 carbon compound which is glycolate leave the chloroplast. So glycolate will exit the chloroplast, oxidize in the peroxisome and metabolize 2 carbon dioxide in the mitochondria. Okay. So for kenapa kita panggil dia sebagai photorespiration? Sebab dia occur during daylight. Okay. So requires oxygen and also organic fuel. Tadilah yang RUBP tu kan bergabung, so dia akan pecah dan jadi glycolate semua tu kan. So it requires organic fuel. And then the process produce carbon dioxide and water. Does not produce any ATP and does not produce any sugar. So this will reduce photosynthesis efficiency, reduce the production of sugar. Okay. So flowering plants have evolved 2 different mechanisms to avoid this wasteful photorespiration, which is a C4 pathway and a CHEM pathway, crassulacean acid metabolism. Okay. So for C4 plant, So these are plants that live in hot dry areas. So pernah juga keluar dalam exam, dia tanya contoh-contoh C4 plant. Maize, okay, maize tu jagung, sorghum, sugarcane, millet. So these are plants that can live in hot areas. Sebab apa? Sebab dia ada adaptation. Adaptation dia ada apa? Dia ada carbon fixation twice. Okay, twice sebab apa? Dia ada enzim PEP carboxylase. Okay, so so bila carbon combine dengan the first carbon dioxide acceptor, which is PEP, so dia akan menghasilkan 4 carbon compound which is oxaloacetate. So that's why kita namakan dia sebagai C4 plant, ataupun for this plant dia undergo C4 pathway. Sebab apa? The first product of first carbon fixation is a 4 carbon compound which is oxaloacetate. Saya short form dia sebagai OAA selepas ni OAA, oxaloacetate. Okay, so for C4 plant awak ingat dia kena ada two different types of cell, mesophyll cell dan juga bundle sheath cell. Untuk C4 plant, ingat dia undergo carbon fixation twice. Ingat ni, twice. Okay, the first carbon fixation akan berlaku dalam mesophyll cell. First carbon fixation akan berlaku dalam mesophyll cell, dan dalam mesophyll cell ni akan terhasil the first product of carbon fixation which is oxaloacetate. Okay, so bila dah terhasil oxaloacetate ni akan convert jadi malit. So malit akan enter bundle sheath cell. Okay, bundle sheath cell is where the second carbon fixation occur. Second carbon fixation ni sebenarnya adalah Calvin cycle. Okay, so ingat Calvin C4 plant ni akan tetap menggunakan RUBP sebagai carbon dioxide acceptor. Dia akan tetap menggunakan Rubisco. Cuma dia ada tambahan ekstra ni. First carbon fixation by first carbon dioxide acceptor which is PEP, phosphoenolpyruvate, by the enzyme PEP carboxylase. Okay, so sebab tu twice carbon fixation. First by PEP, second by RUBP. Enzim untuk yang first adalah PEP carboxylase, enzim untuk Calvin cycle adalah Rubisco. Ingat tu. Okay. So disebabkan oleh bundle sheath adalah site of Calvin cycle, bundle sheath yang warna biru ni, okay, so dia akan menghasilkan sugar. So that's why dia kena dekat dengan vein. Okay, so the sugar will enter into the vein, okay, into vein to be distributed to other parts of the plant. Okay, so inilah dia punya pathway. Saya just explain daripada graph ni, daripada gambar. Untuk C4 pathway, dia melibatkan dua sel kan. First adalah mesophyll cell. So mesophyll cell adalah tempat pertama first carbon fixation occur. Carbon dioxide combine dengan phosphoenolpyruvate ataupun PEP. This is the first carbon dioxide acceptor. So bila dia combine, dia akan hasilkan oxaloacetate which is a 4 carbon compound. That's why kita namakan dia sebagai C4 pathway for C4 plants. And then oxaloacetate akan convert jadi malate. Okay, so the reaction requires an ADPH. Okay, so malate akan enter into bundle sheath cell melalui plasmodesmata. Okay, so bila malate enter into bundle sheath cell, dia akan split jadi dua molekul. Satu jadi carbon dioxide, satu jadi pyruvate. Okay, so kita tengok pyruvate dulu. Pyruvate akan masuk balik ke dalam mesophyll cell. Okay, and then dia akan regenerate balik PEP menggunakan ATP. Okay, so carbon dioxide akan masuk ke dalam Calvin cycle. Sebab apa? Bila proses pecahan malate kepada carbon dioxide and pyruvate, so now you have a high concentration of carbon dioxide in the bundle sheath cell that can enter into the normal Calvin cycle to produce sugar. Okay, so C4 pathway basically dia akan undergo juga Calvin cycle untuk menghasilkan sugar. Cuma proses dia lambat sikit. Okay, sebab dia nak kena undergo carbon fixation twice. Okay, so that is the whole process of C4 lah basically. Ingat kalau C4 plant 2 kali carbon fixation, sama juga dengan chem plant. 2 kali carbon fixation. So graf table ni dia membezakan antara C3 dengan C4 lah kan. So kalau C3 hanya menggunakan RUBP tapi C4 akan guna 2 carbon dioxide acceptor, pertama by PEP, second by RUBP. Carbon fixation kalau untuk C3 once, kalau untuk C4 twice. First compound adalah PGA untuk C3, kalau C4 adalah oxaloacetate which is a 4 carbon compound. That's why kita namakan sebagai C4 pathway by C4 plant. Okay, so enzyme untuk C3 hanya RUBP carboxylase, tapi kalau C4 plant first daripada PEP carboxylase, second RUBP carboxylase. Ini masa Calvin cycle. Ini masa first carbon fixation dalam mesophyll cell. Ini dalam bundle sheath cell. Okay, bundle sheath cell. Alright. Okay, chem plant sama juga. Chem plant macam C4, dia adalah adaptation tapi untuk tumbuhan yang memang duduk kat tempat yang sangat panas lah, dekat desert. Okay, so maksudnya for chem plant memang dia punya stomata terus tutup pada waktu siang. Okay, so kalau macam C4 plant ni, dia akan tutup stomata pada waktu siang. Stomata akan open at night. Okay, bila at night, kalau tengok kat gambar ni, kalau chem pathway kita namakan sebagai crustacean acid metabolism. Okay, so untuk chem plant ni, dia carbon fixation twice juga. Tapi bezanya dengan C4 adalah for chem plant, dia hanya akan berlaku dalam cell sahaja. First carbon fixation and second carbon fixation takes place in the same cell, mesophyll cell. Okay, tapi beza dia adalah kalau untuk chem plant, first carbon fixation berlaku at night. Okay, second carbon fixation berlaku during daytime. Yeah, so basically second carbon fixation ni adalah Calvin cycle. Kan saya cakap tadi, Calvin cycle perlu berlaku pada waktu siang sebab dia perlukan product of light reaction. Okay, so kalau tengok ni yang the blue area ni ya, okay, is when first carbon fixation occurs. Sama je dia punya reaction dia sama dengan C4 pathway, which is carbon dioxide masuk melalui stomata yang terbuka pada waktu malam, gabung dengan PEP which is first carbon dioxide acceptor, catalyzed by the enzyme PEP carboxylase, produce oxaloacetate which is a 4 carbon compound, convert into malate, stored in the vacuole during night time as malic acid. Bila during daytime, malic acid akan keluar daripada vacuole jadi malate pecah jadi carbon dioxide dan juga pyruvate. Pyruvate digenerate balik PEP. Okay, so during daytime, bila carbon dioxide dah terhasil dalam sel, walaupun stomata tutup kat sini kan, tapi still akan boleh terhasil carbon dioxide dalam leaf sebab malate tu dia dah break down into carbon dioxide and pyruvate, and carbon dioxide can fit into the Calvin cycle. The normal Calvin cycle uses Rubisco, uses RUBP to produce sugar. Faham? Itu beza antara chem dengan C4. Perbezaan dari segi masa. Okay, first carbon fixation night time, second carbon fixation daytime. Tapi kalau C4 both akan berlaku pada waktu siang. Cuma berbezanya adalah tempat. Okay, first carbon fixation for C4 dekat mesophyll, second dekat bundle sheath. So ini adalah, kalau yang ni dia punya perbezaan lah kan. Kalau C4 ni, C4 dengan chem plant. C4 dari segi tempat dia, spatial. Okay, first carbon fixation dengan second carbon fixation berlaku pada dalam sel yang berbeza. Mesophyll cell first, bundle sheath cell second. Bundle sheath cell adalah berlaku Calvin cycle. Kalau chem, first carbon fixation during night time, second during daytime. Second adalah Calvin cycle. Okay, so ini samalah gambar rajah yang menunjukkan perbezaan antara C4 dengan chem. Okay, C4 first dekat mesophyll, second dekat bundle sheath. Chem, first carbon fixation during night time, daytime is the normal Calvin cycle which is the second carbon fixation. So ingat basically semua plant, tak kisahlah ada C3 ke, ada C4 ke, chem ke, tetap akan akan undergo Calvin cycle untuk menghasilkan sugar. Maksudnya semua plant ni akan juga menggunakan Rubisco dan juga RUPP. Cuma tambahan untuk kedua ni yang dengan situ adalah dia ada extra carbon fixation, PEP dan juga phosphoenol pyruvate carboxylase as the enzyme. Okay, so that's the end of photosynthesis lah. Boleh faham?
Boleh, madam. Boleh, madam.
So hari tu saya tengok bila saya ada bagi kuis kat kelas lain, punya pemahaman yang salah adalah bila bezakan C4 dengan CAM, dia tak guna dah RUBP, dia tak guna dah Rubisco. Salah ya. CAM, C4 dengan C3 tetap akan menggunakan RUBP dengan Rubisco sebab dia akan tetap undergo Calvin cycle. Boleh So, ada soalan?
So far tak ada, madam.
Tak ada So kalau tak ada, saya harap awak faham. Kalau tak faham, awak boleh tanya kat saya. So ini ada attendance. Okay, you can sign the attendance and then you can leave lah. Okay, kalau tak ada apa-apa soalan, terima kasih kelas.
Thank you, madam.
Okay,
Terima kasih, madam.
right.